Hackett, and she's with the Biomathematics and Statistics group in Scotland, and she's located at the Scottish Crops Research Institute. And Christine has been a developer of this tetraploid mapping software, and we thought that this was a great opportunity to bring her in because of the direction that we're trying to move with our mapping work. So it's a great opportunity to have her here, and I think she's a great opportunity for her to be here in Oregon and tour a little bit, right? So
tour a little bit.
Well, I think we're this one's gonna run past 5 o'clock. So if you feel like you need to leave early, just try to leave quietly, and we'll keep going until we're done.
Can I minimize this? Okay, thank you very much for the invitation to be here. Can people at the back of the room hear me? If not, please. Okay, louder. Okay, right. Linkage analysis and QTL mapping in autotetraploid species. Advanced slide. Any idea why this isn't advanced? The outline of the talk I'm going to give today. First of all, I'm going to give a short theoretical session on the theory that you need to know for using tetraploid map. Then I'll demonstrate some features of the program, and then we'll break for a little while so that you can all try out the exercises in the practical session. During that time, I'll walk around and talk to people individually, and then sum up questions that have been asked that are relevant for everybody. Then in part 2, I'll do the same for the QTL analysis part, a theory section, and then a chance for you to all try out the exercises. So to talk about what we're going to cover on the segregation analysis and linkage analysis, these are all the steps that are carried out if you're doing QTL analysis or linkage mapping in a diploid species. Can I just have a quick show of hands? Who's familiar with linkage analysis and QTL mapping in diploids? Right, that's great, most people. Okay, so these steps will be familiar, but possibly quite a lot of them are done so quickly in the diploid that you don't really register them in the same way. So the first step that we do is a segregation analysis. So that's looking at the parental phenotypes, the offspring phenotypes, and identifying the most probable parental genotype. Then when we've done that, we would do a cluster analysis to partition the markers into the linkage groups. Then finally, within each linkage group, we'll do a linkage analysis. We estimate the most likely phase for each pair of markers and the recombination frequency and the LOD score between them. We then use that information to order the markers, and then we establish the marker phases for the complete linkage group because we need that to do any QTL mapping. So before we can start talking about segregation analysis, we need to have a model for the formation of gametes in an autotetraploid species. And the model that we'll be using is a simple model, random chromosomal segregation, which fortunately fits the data that we've seen on potato extremely well. So we assume that you have 4 parents. Here are the 4 chromosomes of a linkage group. In random chromosomal segregation, we assume that these pair at random to form 2 bivalents, and that each of these 3 possible pairings are equally likely. So you can have chromosome 1 pairing with 2, and chromosome 3 pairing with 4, or chromosome 1 could pair with 3, or 1 with 4, the other way around, to give, in each case, 2 bivalents. We then assume that recombination takes place within each of those bivalents to give chromosomes with the crossovers. And then we assume that 1 recombined chromosome from each of the bivalents is passed to the gamete, so that the gametes that we inherit would look something like that, or that, a combination like that. So in terms of the probabilities, it means that if you have a parent the way you can observe 4 different alleles at a marker, A, B, C, and D, what you expect is that they will produce gametes, 6 different types. So you can have AB, AC, AD, BC, BD, or CD, all with equal probabilities. There is also the possibility that there could be something more complicated going on, that quadrivalents formed and that double reduction products are there, which in practice shows itself as a small probability of gametes with a configuration such as AA or BB and so on. What tetraploid map does is to do a test for this at the beginning, but we hope that this is absent, as the linkage model assumes that there's no double reduction. Again, in the data set we've worked on, I've seen very little evidence for this being a problem. So if you had a parent A, B, C, D, and you were cross that with a parent that had got another 4 different alleles, E, F, G, H, then you could obtain up to 36 different offspring genotypes, even without double reduction. It goes up to 100 if you have double reduction. So it is quite complicated just in terms of all the combinations that you can observe. In practice, we tend to see a lot fewer alleles than this, but you do also have the complication that there may be null alleles at a marker. So the building blocks that I think of are the model, the different segregation ratios that you would have either for a dominant marker like an AFLP or a DART marker. So if we look at the possible segregation ratios for these, then if the marker is present in one parent but absent in the other, then the possibilities are you can have a single copy present in one parent. That's called a simplex marker. And when you cross these 2 parents, that marker segregates in a 1-to-1 ratio in the offspring. And this is quite an informative type of marker. Another possibility is that you have 2 copies of the marker present in that parent. And in that case, when you cross that with the parent without the marker, you'd expect to see a 5-to-1 presence to absence ratio. That's known as a duplex marker. If one parent that has the marker has 3 or 4 copies, then unless there's double reduction, then it will be present in all of the offspring, and that's totally uninformative for linkage. The other possibilities are that an allele is present in both parents. If it's present with a single copy in both parents, then you expect that to segregate in a 3-to-1 ratio in the offspring. I'll call that a double simplex marker. Other possibilities are that there are 2 alleles in one parent and 1 in the other, which gives an 11-to-1 ratio, or 2 copies of the marker in both parents, which will give you a 35-to-1 ratio. Those latter 2, those ratios are too high. They're really not informative for linkage mapping. So that's how you'd look at a dominant marker. But much more informative are if we have a codominant marker such as an SSR. But for these, analysing the segregation ratio to infer what the parental genotype will be is more complicated. So while tetraploid does all this, as soon as you read the data in, it's important to understand what's going on. So we'll look at one concrete example as an SSR that was scored on our potato population at SCRI. So this is an SSR for which there were 4 bands scored. In parent 1, and the bands I'll call them A, B and C were all present. In parent 2, 2 bands were present, B and D. So if you think about what the possibilities for the parents are, if there are no nulls involved, then in parent 1 there must be a double copy of one of those alleles, but we don't know which one. So the possibilities are AABC, ABBC, or ABCC. Or it could be that there's a null present and that parental genotype is ABC null. Or the parent 2, there are 2 markers, so there can be 3 of one and 1 of the other, 2 of one and 2 of the other, or there can be 1 or 2 nulls. So on the basis of the parental phenotypes, we can say that these genotypes are possible. If we look at the individual segregation ratios of the markers in the offspring, then you can get a clue about the most likely genotype. So, for instance, in this population, A was segregating in 167 to 22 presence to absence ratio. That's closest to 5 to 1. So it's most likely that that is a duplex allele. Similarly, B, segregating 149 to 40. That's present in both parents. So among the ratios for marker present in both parents, that's closest to a 3 to 1 marker. C is close to a 1 to 1 marker. And D, again, is closest to a 5 to 1 marker, but present in the second parent. So on the basis of this, we've hypothesized that our cross is AABC by BDD null. But that's on the basis of looking at each marker separately. What we want to know is that consistent if we look at all the bands together? Or are any other configurations more likely? Tetraploid map uses a result called Bayes' theorem to calculate the probabilities of each of the possible genotypes. But what it's doing really is to compare the observed band patterns and their frequencies with what you would expect from the different configurations. So, for instance, when we looked at how many offspring had the band pattern A, B, and D, there were 72 compared with these expected ones under 2 possible models. And what we found for this marker was that the configuration that we expected from looking at the bands separately was actually a very good fit to the data, a chi-squared value of 10 on 7 degrees of freedom. It was a good fit to the data. Compared with the next most likely configuration, had a chi-squared of 47, again on 7 degrees of freedom, which would be a poor fit. You would reject that. So the probability argument by Bayes' theorem shows that this configuration was by far the most likely, and we would use that. If tetraploid map really can't tell what the configuration is, it won't carry on and use that marker. So that's the steps that's carrying on in the cluster, in the segregation analysis. After that, there's a cluster analysis. You don't really see the first cluster analysis because that's done immediately that you read the data in. But what that stage is doing is to look at the simplex markers only and to cluster them according to whether they're linked in coupling on the same chromosome. And it will use a code which follows through that marker all the way through. These simplex markers linked in coupling are particularly informative about the markers. Later on, you'll do a manual cluster analysis to actually work out where the linkage groups are. In that case, the cluster analysis is based on whether the markers are segregating independently or not. Hi. Sorry, you still can't hear me? Okay, I'll try. The second stage does a chi-square test for independent segregation, and the more significantly associated the markers are, the closer they are in the cluster analysis. Right. I'll pass on the dendrogram until we actually demonstrate the software. In terms of the linkage analysis, I'm only going to cover this quite briefly because I know that time is short. To set the scene in terms of the configuration that you'll be familiar with from analysis of diploids, if we assume that there's no double reduction and look at the recombination frequencies among 3 simplex markers. So I have A and B that are both situated on one chromosome here, C that's situated on another chromosome of that linkage group. So if we wanted to calculate a recombination frequency between A and B, then just as in a diploid, we would look at a table of A present, absent, and the recombination frequency is estimated as the total in the off-diagonal classes N2 plus N3 over the total number of genotypes scored N. So if the markers are in coupling phase like A and B, that's fine. If the markers are in a repulsion phase like A or B with C, then calculating the recombination frequency like that will give you a recombination frequency greater than 0.5. So that signals a repulsion linkage. Again, You've seen this if you work with diploid outbreeders. For a diploid, then in that case you'd just calculate the recombination frequency as N1 plus N4 over the total number. The equation changes for repulsion in a tetraploid species. The recombination frequency is actually estimated as 3 into N1 plus N4 over N minus 1 in that case. And one important thing to know about a linkage is that this type of linkage, simplex markers and repulsion, is a very unreliable linkage. That's associated with an extremely large error. So you normally can't use that type of marker for establishing linkage among homologous chromosomes. So much more informative situation is if we have 8 different alleles or more than that, where we can pull all of the linkage groups together. I'm not going to go into details of tetraploids algorithm for looking at that. It's based on looking at the number of recombinants and counting those and estimating those for different configurations. But one important thing to remember all the time is the phase. If you're working with SSRs, then you're not going to know which alleles are linked in coupling on which chromosome. Tetraploid map will compare all of the different phases and select the most likely phase for each pair of markers. So once that 2-point analysis has been done, so we've found out the most likely phase, the recombination frequency and the LOD score between all pairs of markers, then tetraploid map will go on to order those. It orders using a weighted least squares criterion, which is the same criterion that was used by JoinMap version 3. It has 3 different options for ordering the markers. It always starts with an algorithm based on seriation, which is a fast way to find a possible initial configuration. After that, it will then try a ripple search, which is fairly fast. And then finally, there's an option of doing a search based on a more general search algorithm called simulated annealing. What we would recommend is simulated annealing is slow, then stay with the ripple search for most of the time. When you initially cluster, you may decide that there is a marker that doesn't actually belong in that linkage group. The ripple search is enough to show that. Only when you're sure that you're coming to a final set of markers that belong together is it worth spending the time to do a search with simulated annealing. You're talking about in tetraploid map. This is a question about using simulated annealing with a large number of markers, and the software may crash. Yes, I think there's an upper limit of 40 markers for using simulated annealing. So if you're above that, then you'll either have to remove a couple of closely linked markers or stay with the ripple search. Yeah, it does get incredibly slow for larger numbers. Mm-hmm. Yep. And then hopefully towards the end of this, then you will get a list of the linked markers, their positions, and enough information to be able to infer which marker allele belongs on which of the homologous chromosomes, which you'll need for the QTL analysis. Right. So I'm going to start demonstrating the analysis in a minute. I just want to say a little bit about the layout of the file. The input file in the potato.loc. On the first line, you show the numbers of individuals present, 228, and the number of markers, 73 in this case. Then you have the marker name followed by the number of alleles. So first few of these are AFLPs, so there's a single allele. You score it as present or absence, 1 or 0, with a 9 for missing values. The first 2 numbers, that shows the presence or absence in the parents. After that, then you've got presence or absence in the offspring. So this 228 that's the number of offspring only, so that there will be 230 numbers on the line, including the parents. Further down here, we've got an example of an SSR marker. In that case, there are 4 alleles, and there are 4 lines here representing presence and absence of the 4 different SSR alleles. So these are scored as, so you've got alleles 1, 2, and 4 present in the first parent, alleles 2 and 3 present in the second. Something that's very important as regards missing data in SSRs. Tetraploid map can't use a data that's marker that's got partially missing values for any individual. It needs to be either all present or all absent. If there's a 9 in the first row of an SSR, it will take that as missing. What it can't deal with is a 0 or a 1 in a first row and a 9 somewhere else. So it's probably safer to mark 9s all the way through. So that's the form of the data. Right, I'm going to escape and demonstrate it with Tetraploid map now. I suggest that you probably watch me for now and then try when I break. So the Tetraploid map's been put in all programs. And what we do is to normally start a new project by clicking here and browse for the location, and I'll give this a new name. So that creates the project and asks you to import data sets, which you do by this import. It's not seeming to find the data. Okay. The potato.lock is the data. So this has both read the data in and done quite a lot of the preliminary segregation analysis. So to go through the columns, we've got 73 markers read in here. You've got the names in this column here. At the moment, they're all selected for use. Right. The type is given as either AFLP or SSR. AFLP is shorthand for dominant marker with a single allele, so it can't tell whether it's AFLP or DART or something similar. Then you have the number of alleles next to it, which is 1 here, 4 for this SSR. And you have the marker phenotype. This is the phenotype of the parent 1. So that's given as present here, and then of the 4 alleles, 1, 2 and 4 are present for this SSR. And you've got the same information for parent 2. Right. I should have said this is a cut down version of the data from the SSR potato population Sterling by 12601, and it consists of some data markers from Sterling parent plus a few SSRs. So that's why we're not finding any or very few markers marked as present in parent 2 in this case. So you've got the parent information. Then this starts on the analysis that's been done. It's inferred a genotype and what the ratio is, whether it's a 1:1, a 3:1, a 5:1, or so on. That's in the column headed ratio. Maximize that a bit. Then you've got a ratio sig column. The smaller that is, the more significant the distortion of the marker from the expected segregation ratio. And then finally, we've got 2 columns related to double reduction, which in the interest of time, I'll probably skip over. If anyone wants to ask me more about that, come back to it. I didn't talk about the first column. This is the one where the codes have been put in to show markers that are simplex markers linked in coupling. So we've got a group here of markers that are labelled AA_W_T, about 5 markers with the same code. And that's given us a clue immediately that those markers have been found to be likely to be simplex markers linked in coupling on the same chromosome. The actual code is you've got 3 parts separated by underscores. The right-hand one, the letters are the same if the markers are linked together with a recombination frequency of 0.35 or less. So the second code is, are the markers linked together with a recombination frequency of 0.3 or less? The third is, are the markers linked with a recombination frequency of 0.25 or less? So if the code is completely the same, the markers are identical with a recombination frequency of 0.25. If I scroll down, and I found an example. Yes, you've got 3 markers here, G and K. So those 3 are linked with a recombination frequency of less than 0.25. But up above it here, you've got one labelled FNK. So that's likely to be in the same chromosome, but it's a little bit further apart. It's between 0.25 and 0.3, as the last 2 letters are the same, but not the first one. Okay, if we want to click on any marker, mark here, then we have summary information appearing at the bottom. So this is giving the phenotype in the parent, so present in parent 1, absent in the other parent. There's a test for double reduction, which isn't really of any interest in the dominant marker like this. Then you've got the different band patterns, the marker present or absent. You've got the numbers and the ratios. And then you've got the probabilities of the different configurations. So if we look at the bottom here, in the absence of double reduction, which is what we're most interested in, then the program is comparing the possible configurations of a simplex marker or a duplex marker. So this is the simplex marker with the single 1. That's been given a probability of 1. This is the chi-squared statistic testing that and the significance of the chi-squared. Compared with a duplex marker, posterior probability for this of 0. The chi-squared is 188, and that is rejected in favour of a 1-to-1 marker. So something else that you can look at, if you look at parent 1 linkages here, then that will summarise tentative linkages between simplex markers and duplex markers to help you think about where the recombination frequencies would be. So this was a simplex marker here. It's identified a duplex marker here that it's probably linked to. The C in the phase column shows that that's a linkage in the coupling phase. And you have the chi-squared test for those segregating independently and the significance of that, which you see is very low. Those are strongly linked markers. So that's all the preliminary information that you get when tetraploid map reads in the data. The next thing is to select the markers that you want to use for the further analysis. At the moment, they're all selected. So we've got this dialog box to select the markers. So what I would normally do is to start with the AFLPs, and here we have markers from parents 1 only. Select the markers that are a good fit to a 1-to-1 ratio. So I'm using a significant threshold of less than 0.001. So I'd select those, and I'm told 59 markers of the 73 fit in that criterion. Now if I also select the 5-to-1 markers, the 5-to-1s I'm going to be a bit more stringent. If you have a distorted 5-to-1 marker, it can have a lot more impact on your map than a distorted 1-to-1 marker. So I tend to be more stringent in my choice of duplex markers. And as I want to select these in addition to the 1-to-1 markers I've already selected, I check that here. And then finally, I would want to select the SSRs. So that selects most of the markers. Can I drag this somewhere else? Right, it's not scrolling very well. Right, so once you've selected your markers, then you can go in and cluster them to look for linkage groups. Tetraploid map was set up for potato. It does give a default of 12. As I know that this is a subset, I'm going to choose 6 groups. But in the first case, what we want to do is to inspect the dendrograms to see how the markers are splitting. Tetraploid map gives you 2 different clustering algorithms, a single linkage clustering and an average linkage clustering. And there's a bar here that you can draw across, click on it, and at any point you'll be told how many cluster groups are formed, and the colours show how the markers split up. So we can compare groups. At that level you'd have 6 groups forming, and you can look and see how the markers occur like that. This is one of the hardest bits. It's more subjective. I think it's the same in the diploids, exactly where you cut the markers into the linkage groups. If you form a too large a linkage group, then it's easy. You can just go back and highlight that particular linkage group and cluster again and see where the markers split again. If we decide to stay with 6, then we can look at one of the linkage groups. It shows the markers in that, and we can then go ahead and order these markers. So at the moment I'm using the first 3, including a ripple search, but not the rest. And that runs the ordering. And then in the details you have the markers in order. So if you click on any marker here, then it will show a list of all the other markers and their recombination frequencies and their LOD scores with the markers selected on this side, and panels at the bottom showing the recombination frequency and the LOD scores as well. So if you want to know then more information about any particular pair, then you can click on the marker on this side, and then this opens up a dialog box at the bottom which has some information about the phase. So if you think of each of these pairs of numbers vertically as representing the markers along the chromosome, then what that is saying is that this first marker is a 5-to-1. It has the 2 alleles which have been randomly allocated to chromosomes 1 and 2. However, this marker next to it is linked in repulsion phase. The 1 there is on a different chromosome to the 1s and 2s. If you compare that with another marker, so that is a marker 298, that's labelled on the chromosome with the code G and K. That's linked in repulsion with that duplex marker. If we look at this one, the label BXU, and you see that the code has switched so that now the allele for the simplex mark is on the same chromosome as one of the alleles for the duplex mark. So that's showing a coupling linkage. And if we want to look at this as a map, press map, choose which parent to use, and that shows the overall markers. Want to put in phase information, if you need to, then we just have to type that in here. So that's a 111 note. When we looked at the phase of that with this, then that was on either the 3rd or 4th chromosome. Randomly say it's on the 3rd. And then everything else that's labelled with the G and K code would also have that same phase. This BN here is on, that's on the same linkage group, so we would put that as a code 1.0, and so on, and you fill that in for the entire linkage group, just to show the partly filled one. Then it started to fill in which markers belong on which chromosome. There are buttons here to save the linkage map either as a graphics file or as a text file, and this is saved in a format that can be easily taken into software such as map chart. Okay, right, I'm going to stop talking now and let you try out part one of the exercises in the practical session. So we're starting again for people who are listening to the webinar in about 15 minutes, I think. Okay, we're going to start again now and talk about some QTL analysis. In the meantime, I want to answer a question that was asked during the break, namely, how do you get hold of tetraploid map and how much is it? The answer is you can get it from the BIOS website. The fastest way to do that is to just put tetraploid map into Google and it will probably come up first. In terms of cost, it's free. It was developed under a grant from BBSRC in the UK. The terms and conditions were that the results had to be made freely available, so there is absolutely no charge. And there is a file that you download with it, something like tetraploid map for Windows.doc, which is quite a detailed manual with a tutorial, and there's some demonstration data, which is simulated data, so slightly better behaved than this. So if you want to try it out, that's how to get hold of it. So to carry on now with some QTL analysis. If you want to locate a QTL in a population like this, again, the easiest way to start is a way that you'd probably start in a diploid population, and that's to do analysis of variance on each marker in turn to see where there are significant differences between the presence and absence of the markers. So tetraploid maps has got two options for doing that. You can either do the regular analysis of variance, or there's a Kroskal-Wallis test, which is a non-parametric analysis of variance, which is perhaps more valid if your data isn't strictly normally distributed. So the analysis of variance option of tetraploid map will do both of those for you. This is a useful first scan. It's not completely informative because the different markers may have so much different information about what's going on. If you look at a diagram like this, if that's the chromosome, then you have a QTL in the position here, and next to that you have a simplex marker that's present in parent 1 only, and then you have next to it a fully informative marker with 8 different alleles. If your QTL is actually only segregating in parent 2, then this marker, although it's closer, it won't give you any information about the QTL. So you'll only get information in terms of the more distant marker that is segregating in the same parent. So this analysis of variance, it gives you a clue as to which chromosomes are the most important, but it doesn't give you the full information. So what we need to do is to use marker information for the whole chromosome to start inferring QTL genotypes at a grid of positions along the chromosome, and then relate those to the trait information. And again, this is just what you do in the diploid species, just the process of inferring the QTL genotypes is a bit more complicated. So we can't observe a QTL genotype. All we can do is put together a model. We'll assume that there are 8 different QTL alleles. We'll call them Q1, Q2, Q3, Q4 in the first parent, and then Q5 to Q8 in the second parent. And from those we can write offspring genotypes in a form like this, Q1, Q4 from the first parent, Q5, Q6 from the other parent. And you can theoretically get into very complicated models with a lot of interactions. This is the equation that I swiped, includes the main effects, interactions between pairs and triples and quadruples and markers. You could in theory fit all that, but in general you don't normally have enough information. What we tend to look at in practice is also to say we normally don't have enough markers to be really sure of our alignment of the maps in the 2 parents. So in terms of QTL mapping, we tend at the moment to work in terms of each parent separately. So for each parent separately, that gives 6 different QTL genotypes. So we fit a full model where we fit a different mean for each of the QTL genotypes. And we'll compare that with a set of simpler models, either of a simplex allele segregating or a dominant duplex allele. So I'm going to just go fairly quickly through the reconstruction of the offspring. There's a search algorithm going on to look to see how could the offspring genotype be obtained from the parents with as few recombinations as possible. And the result of that search algorithm is a graphical genotype, something like this. So this was one particular offspring from the set. This was the phenotype of this offspring. And we found out that if you look to say how can this actually be obtained, then we find out the way this happens is if chromosome 1 and chromosome 3 paired in the first parent and there was a recombination at this point here. Chromosome 2 paired with chromosome 4 in the first parent and a recombination here. Chromosome 6 paired with 7 in the second parent and there was a recombination somewhere in that region, but there was some uncertainty here as to which side of marker L8 it is. Or there's this chromosome, there had to be 3 crossovers, and we're slightly uncertain about 2 of the regions. So that's that's a reconstructed graphical genotype for this offspring. And then we could go on to put probabilities on that. So in this area, there's only one possible QTL genotype. This area is at a crossing. There are 2 possible QTL genotypes, and we change the probabilities according to distances from the markers. This area, there are 2 recombinant areas, giving you 4 possible genotypes in this area. And again, tetraploid map gives different probabilities to this. And then in actually calculate the likelihood profile, it will step along the chromosome, relating the trait data to the inferred QTL information at that point, and changing the weights in a weighted regression. We're using information on all markers. We use information on all markers to infer what the most likely QTL genotype is. Typically, there isn't enough information if you only use flanking markers. I mean, even a program like MapQTL tends to use five markers each side, and you can change that. But we use all the information on the markers in a chromosome. And you finally come out at the end with a LOD profile. Right. In terms of what the trait data looks like, again, it's a simple text file. You have the number of traits that you're looking at the top. Then there's an index down the side numbering from 1 to your total number of individuals. Yes, the Haldane's mapping function is being used to order the... Sorry? The question was, does the mapping function change from diploids to tetraploids? We've never really done anything to check that out, to be honest. We just assumed Haldane's. Again, my impression is that even in diploids, people may use Casambi's function for looking at the map. But if you look at the theory that's going on behind the QTL mapping, it always assumes that recombination in adjacent segments is independent of each other. So in effect, Haldane's is being assumed for the QTL mapping, even if the map's been put together with Casambi. In terms of the trait data, then you've got the names of the traits here. Here we have data set on maturity and tuber blight percentage. The numbers are here. The missing value code is -99.0. So I'll switch over now to tetraploid map. Right. What I'm going to do is to start again, and this time I'm going to load the potato.proj file that you should all have. I'd suggest that when you start again with that. because in that project all of the phase information has been filled in correctly. That's one of the longest stages of using tetraploid map. So, potato.proj. This is exactly as we had before, but with the phase information filled in. So, for instance, if I look at a linkage map like this is the linkage map of chromosome 5, and all of the phase information has been filled in. So, to load the marker data, if you go to markers, there's a menu that says associate trait data, and there should be a potato.qua file to open here. That names the two traits. We want to use both of those. So the first step is to do analysis of variance. That's the ANOVA button here. Right. So what I'll do now is to concentrate on the maturity data, and then when I've finished I'll have another pause and you can look at the tuber blind.
I've got a question from the webinar people, and how many markers can be used to make a map? We have 9,000 snips potentially. How much can the program handle right now?
The question is how many markers can tetraploid handle right now, and somebody's mentioned 9,000 SNPs. At the moment it has a limit of about 800 markers. We've been working with larger data sets than that, but we've tended to do some pre-processing in another program to eliminate duplicate markers. Also, I would probably want to work with each parent in turn, so you would cut down the markers to some extent. We're very interested in hearing from people that there's the need to develop tetraploid map for larger projects, though. So this is something that we clearly need to build a new version for. So when you've done the analysis of variance, you end up with this panel at the top. If you click on maturity, then that tells you which markers are showing any significant association with maturity at all. So the criterion is, it's done two types of analysis of variance. The AV significance is the usual parametric analysis of variance. The KW significance is the non-parametric Kroskal-Wallis analysis of variance. If either of those has a significance less than 0.05, it will be listed here. Of course, we're most interested in the most highly significant markers. And what we see here is that there are a group of markers from the chromosome labelled AAWT that are all showing very high significances associated with maturity. So the columns that you have here are the significances, the mean trait value in the absence of the marker, which is 5.6, the mean trait value in the presence of the marker, which is 4.0, and a standard error of the difference between the two. So this would show this as a linkage group that we're most interested in starting with. So this is linkage group 5. So if we go into that and click on details, then we can run the QTL analysis. The QTL button doesn't highlight until all of the phase information is in correctly. Once that's in, we can perform QTL analysis. It will ask which parent. So we choose parent 1. The moment it's going through phase 1, so this is inferring what the genotypes at each individual are. Then phase 2 steps along the chromosome in 2 centimorgan steps, looking at the LOD profile. So if we look at maturity, we'll see that there's an absolutely enormous QTL maturity in the middle of that chromosome. The LOD score, the maximum LOD scores listed here is 33. So this position is explaining 55% of the variation in maturity. And we have the mean maturity associated with each of the QTL genotypes listed here. So this is fitting 6 different QTL means. You can compare this with a simpler model. So if you look in the simple model, then these are the simpler models that are being compared. There are 4 simpler models of a simplex allele on each of the 4 possible chromosomes, or there are 6 simpler models of a dominant duplex allele. If there's any simpler model that's not significantly different from the full model, it will be shown in bold. So for instance here we have C3 versus the rest shown in bold. And if we go to this green button here, that's to rescan the whole chromosome fitting that simpler model. And the simpler model that you choose will be highlighted here, although you can change that. And now we get, in addition to the red line for the full model, we have the blue line fitting the simpler model. And we can go back here. And we see that this explains 55%, but the simpler model is explaining 54% of the trait variance, just by modelling that there is a simplex allele on chromosome 3 that's affecting maturity. Please note there's a typing mistake in here. That should be plus C3 or minus C3. It shouldn't be minus C4. So if you look at the overall genotype means for each of the combinations, that makes sense. And that you see that the combinations involving chromosome 3 all have much lower score for maturity than the combinations that don't. And if you wanted to look at a log threshold for this, then you've got this button here that will run a permutation test. You can either run a permutation test for the full model, or you can run it for the reduced model, which we'll use for the reduced model. This is quite slow. So this is probably a good point for everybody else to start opening up the project, Tetraploid project.
Actually, a manuscript that was accepted for publication a little while back. We run Tetraploid map and did 100 permutations.
Yes.
But we actually got asked to do it on 1,000 permutations. What's your opinion on that? I mean, is it that much robust when you run more permutations, and was it a reasonable request for the degree of precision that you obtain with the 10-fold more permutations?
Okay, the question was about the number of repetitions to use in a permutation test. It's become very popular to ask for 1,000 permutations. In a model like this, it takes a long time. As you say, we're doing 100 here. There was a very nice paper, I think by Nettleton and Dirge a couple of years ago, where they looked at putting confidence limits on a permutation test, so that you could say run 100 and then you could get a range for your confidence limit. So instead of just looking at the 95% point, you'd maybe look at a point a bit below that and say, if your threshold, if your maximum LOD is definitely below that, it's certainly non-significant. If it's above this upper limit, it's definitely significant. If it's in the middle, you need to run some more to make your test more precise. So that's something that I tend to do. I can't remember the exact reference offhand, but I can email it to you. I think Nettleton and Dirge were the authors of that paper. 100 permutations is rather on the low side. I mean, we set this up so that people can copy the 100 permutations into an Excel spreadsheet and then run another 100 permutations again and sort and build it up. That was the line that we were thinking on, if you've got the patience. But
well, it wasn't me who had the patience, somebody else did.
Yeah, I would use a confidence interval like that to see if it was really necessary or not. Yeah. Okay, so the permutation tests run. You end up with a dotted line along the bottom showing the threshold. If you go into LOD details, it gives you the 90% point and the 95% point and all of the individual values in the simulation, in the permutation test. So, as we were just saying, you can cut and paste those and use them elsewhere.
So in your example, let's say the whole chromosome is above the threshold.
Yes, this. this is probably a pretty major gene for maturity here. Right. Okay, I think that's about all that I wanted to say about the QTL mapping. So people can carry on for as long as they have patience to stay, have a look at the tube of light data and the model for that.
So
At this point, for the people that are on the webinar, we're going to close out the webinar, and the people here at in the computer center can stay on and work with things, and Christine will stay here. But I think at this point, first of all, let's thank Christine for her presentation. And then secondly, our webinar people. We've had people from around the world, especially I want to thank our listener from New Zealand and also from the Netherlands. I don't even know what time of day it is to figure it out, but you guys are real troopers that you stuck through this whole thing. And then lastly, there's our behind-the-scenes people. Jeanette Martins has been kind of our conference workshop organizer. She's probably running around in the hallway somewhere. And then over here, sitting in the corner, who is critical to the whole success of this functioning webinar and event has been John McQueen, who is our resident OSU person here. It was just lucky that we happened to have it here in Oregon State this year, and John is our SolCAP PBG Works webmaster, I guess. I don't know how you define yourself. So I'd like to thank those people for really making this thing work today. And then lastly, having Robin Buell, Alan Van Dynes, and Christine Hackett, I think was a great group to come in, and next year we'll have another SolCAP workshop, and the workshop will be based upon what we really think we need for the community. It's not what we think you need; it's what you feel that really is most helpful to you to help you move forward in your use of the SNPs and the potato analyses. Put on the survey. Okay. Thank you.